Okay, hello everyone. My name is Bettina Schirrmeister. I'm from the University of Bristol, where I do my postdoc at the School of Earth Sciences. I'm going to talk to you today about cyanobacteria, how the evolution of cyanobacteria could be related to the Great Oxidation Event. I did look at fossil data, but also at genomic data to reconstruct the evolutionary history of cyanobacteria. So most of you people will work, I guess, on animals or plants or organisms that lived somewhere in the Phanerozoic. Many of those organisms, animals and plants, will rely on mechanisms such as oxygenic photosynthesis or aerobic respiration, both of which have their origins deep down in the Precambrian. So this is here you see a timeline where I highlighted major events of the Precambrian. This would be like formation of continents around 3.8 billion years ago. We have first indications for life around 3.5 billion years in Australia and South Africa. First eukaryotes in the Proterozoic sometime around 1.8 billion years. And finally, as we heard in the previous talk as well, an increase in oxygen levels, a rapid increase referred to as the Great Oxidation Event around 2.4 billion years, which can be taken as evidence for the presence of oxygenic photosynthesis. So, but how do we get an increase in oxygen if we look at it from a biological view? So, I had this slide, but it was covered very well in the talk previously. So if you look at oxygen levels in the history of our planet, we heard that we have those three levels, which might have a lot of fluctuation, also here in the Proterozoic. But we do have an increase at least 100-fold, so from 10 to the minus 3 of present atmospheric levels of oxygen to 10 to the minus 1 here at around 2.4 billion years ago, which should be due to the presence of oxygenic photosynthesis. We might have local oxygen oases already earlier, up to 3 billion years ago. And so where does the free oxygen come from? Oxygenic photosynthesis, cyanobacteria, blue-green algae. They are the ones that produce oxygen during photosynthesis. So that functions, they reduce sunlight, carbon dioxide, to produce organic carbon and release free oxygen. Now, naturally, this free oxygen would be used up at the same time by during aerobic respiration by other microbes, which would use the oxygen and the organic carbon. This happens during decay. So to get an increase of free oxygen, we would need additional carbon burial to get free atmospheric oxygen. Now, cyanobacteria, so we know they do oxygenic photosynthesis. They have been around 2.4 billion years ago. We see this rapid increase in oxygen. What do we know about the fossil record? What can we relate the evolutionary history of cyanobacteria somehow to this increase of oxygen? So if we look at the fossil record, we do see a lot of various types of fossils in the Proterozoic, nice filaments of varying sizes, partly even with really distinct morphological features. We also see a lot of fossils in the Archean Eon, also some filaments, spheres. The good thing about the Proterozoic is that many of those fossils, there's so much characteristic preserved that we can link them to modern taxa, many of which are cyanobacteria, very nicely, as you see here, for example, for spirulina. And we see this also in the name, like they will have similar names, like Palaeontological is an ancestor of Lumbia, presumably. They look very similar. But for many other fossils, including some of the early Proterozoic ones, there's more question regarding the taxonomic identity of those fossils, particularly in the Archean Eon. People have tried to solve this with other methods, for example, also biomarkers, where there have been reports that biomarkers could link cyanobacteria around 2.7 billion years ago. But it turned out those biomarkers were much younger and they're not as characteristic as we may think for cyanobacteria. So we don't know really a lot about taxonomic identities of fossils in the Archean. So the gunflint shirt, this is an early Proterozoic deposit, and there has also been a lot of debate about what sort of taxonomic fossils do we have in the gunflint shirt? What sort of microbial community was there? Were cyanobacteria involved, or was it? People have argued that it might be proteobacteria, leptothrix that are present in this deposit. So the Gunflint is around 1.8 billion years old. It's one of the most impressive fossil deposits we have from the Precambrian. You can get the idea if you look at the variety of fossils we see and the different forms, like umbrella-shaped fossils. Here, several, I didn't show all the filaments and spheres we see that we also find that are quite common. The Gunflint is located here in Canada at Lake Superior. It has first also, it's outstanding because it was the first deposit that has been described where we find microbes from the Precambrian. In 1965, Barkman and Tyler have described fossils from the Gunflint. So we have seen a lot of studies from the Gunflint using microscopic sites. So I was trying to use synchrotron radiation X-ray tomography. This is, here you see again some microscopic slides. You see this is we have a lot of tubes and sheaths here, some larger spheres. Now, it's good. You can use the Gunflint very nicely with synchrotron data because the material of the Gunflint is mostly quartz, and the fossils are carbonaceous remains. So here we have colored this is the Raman spectroscopy, an image. Those are the C bands. They have been colored here in green. The quartz will be in blue. So the back of the rock matrix, and you can. Now, with the synchrotron, using synchrotron X-ray radiation tomographic microscopy, you can basically, you get those images where the rock matrix will have a certain, it has a certain density, and then the carbonaceous matter will have a lighter density. This will be detected, therefore it's colored darkly. We can now color those, reconstruct fossils within the rock matrix. And this is how one of the Gunflint samples looked like. So we see a variety of different morphotypes in this rock matrix here, such as large spheres up here. We have a cluster of smaller cocci or bacteria in this area, and down here we have a lot of filaments of varying sizes. We did find also some other morphotypes which have been reported previously in the literature quite well from microscopic sites, such as star-shaped patterns here, spheres. Those are diplococci, so sort of they seem to be attached. We have filaments of different sizes. And I have a student of mine, Ella Nadian, a previous master student. She has looked through 120 of those rock samples and has tried to look at the abundance of those fossils, so of spheres that are below 8 micrometers or larger than 8 micrometers. We only looked at so we have a wide range of different spheres of different sizes, also filaments, and we only divided them by sizes. We didn't try to identify them taxonomically. But if you count those basically, and also the filaments, we have large filaments with diameters larger than 7 micrometer, and some that have diameters smaller than 7 micrometers. And then those other morphotypes. And if you look at the distribution of those fossils in the Gunflint, we see that we have mostly small spheres, also a lot of large spheres, filaments of varying sizes, and then much less those other morphotypes. Now, with those varying sizes of filaments, I thought maybe we could still make some assumption about what this might have been in the Gunflint, what sort of what those filaments could have belonged to. So if you look at the fossils from the Proterozoic, we see that a lot of the filaments have very strong sizes, vary quite largely, also compared to the Archean fossils here, which are much smaller. Now, if we look at the Gunflint, the filaments in the Gunflint, they have been described as Gunflintia minuta and the larger forms as grandis. So Gunflintia minuta we see by a study by Boll and G from 2010, they have described those as fossils that are below 2 micrometers in cell diameter. So this is here we have cell length, which is not shown for the fossils, and cell width. So because cells are rarely preserved within the filaments, so we only look at diameter. So for the Gunflintia minuta, this would be below 2 micrometers. We do have diameters for Gunflintia grandis below 4 micrometers, 2 to 4 micrometers. And in our study, what we found, the larger filaments, they could be here up to 10 micrometers in size. Now, if we look at modern fossils, modern bacteria, fossils, living bacteria, and cell sizes. So this is, we have filamentous bacteria occur variously in Cyanobacteria and Proteobacteria, Schirrmeister and Bacteroidetes. In all of those modern taxa, we do have filaments. Now, if we plot I did look at average cell sizes here. So this is the length of the cells and this is the width. Cyanobacteria are shown in blue circles, Proteobacteria here in squares, Chloroflexi in triangles and green triangles, and Bacteroidetes in red diamonds. What you see is that Ganflinsia minuta it fits very well size-wise with most of those bacteria. So it could be Chloroflexi, Bacteroidetes. If we go to Ganflinsia grandis already a little bit, it's not it can't represent any filamentous bacteria we know today. Rather, Cyanobacteria and Proteobacteria share sizes with Ganflinsia grandis. If we look at the filaments, the large ones we have described or we have seen in our study, they clearly resemble rather Cyanobacteria or Proteobacteria. Additionally, Proteobacteria will have widths of filaments in Proteobacteria, something like Vegetator, marine Vegetator species. They can get up to 100 microliters. I haven't shown those here because they're way over there somewhere, so they wouldn't fit nicely in this figure. So the larger filaments we see in our study, those ones here, they rather resemble maybe Cyanobacteria or Proteobacteria, such as Vegetator, really large filaments, rather than any other forms. But so now we know in the Ganflin herd there might be Proteobacteria and Cyanobacteria. We don't know exactly how to distinguish between those two. What could we do? Could we take, we could take modern samples maybe, modern data, look at 16S data or something, reconstruct phylogenetic trees. Maybe we could reconstruct the evolution of multicellularity within those taxa we have today. So I tried to do that. I did look at Eubacteria. I tried to look at a wide range of eubacteria and to identify where do we have multicellular forms and how did these multicellular forms evolve within the phylogeny. So first I would like to point out that if I'm talking about multicellular prokaryotes, I'd like to refer to this quote by Robert S. Reed from 1948, where he says that multicellular prokaryotes are filaments which are composed of dividing cells which remain more permanently together and are normally flattened against each other on adjacent sides. So I'm not talking about cell chains that will form as a response to environmental conditions and lose up again, but I'm talking about permanent filaments in those prokaryotes. Now this is the phylogeny I have reconstructed with 16S data, ribosomal gene. Those are 3,500 approximately taxa. I have identified different phyla across this phylogeny, and those would be the ones that where we have multicellular forms. That would be, as we discussed earlier, would be the Chloroflexi, the Bacteroidetes, the Proteobacteria, Cyanobacteria, but also the Actinobacteria, where we have a lot of filaments. Now, the problem here is that at this large phylogeny, we have really weak support for deep nodes, deep branches. So if we go far back in history, it's hard to say how those different phyla relate to each other. So what I did is I subsampled from this huge phylogeny a smaller set. Those are two genes, 16S and 23S, concatenated of 105 taxa that cover most of those phyla. And we see immediately this is a Bayesian phylogeny. And we see immediately that posterior probabilities at deep nodes are much more convincing, so almost close to 1 for all of those. Now, if we only look at the multicellular taxa, again, we have the Proteobacteria, where we have multicellular forms like Vegetator, Leptothrix. Here we have the Bacteroidetes, Cyanobacteria, the Chloroflexi, and the Actinobacteria. Now I did try, could we reconstruct the evolution of those characters? Could we say which ancestor might have been multicellular among those eubacteria? So again, here this is a character state reconstruction using maximum likelihood methods. This has been done over 3,200 trees, which are all equally likely. This is to incorporate phylogenetic uncertainty. The characters here of the present taxa are shown in yellow would be unicellular and black would be multicellular. Now, if we reconstruct those characters, we see that in Proteobacteria and also Bacteroidetes, those filamentous forms we see are recent transitions. It's no multicellular clades where a lot of those taxa are multicellular, but it's just Vegetator as a recent transition, Q-Thrix, Meganema here, and the same for Bacteroidetes. If we look at Cyanobacteria, Chloroflexi, and also Actinobacteria, it seems that multicellularity is much more common in those phyla and occurred probably much earlier than for the Proteobacteria and the Bacteroidetes. This is just a more detailed tree of the proteobacteria, and again we see here the Bettina, Theoblocker, Theothrix. They seem to be, and also Leptothrix, seem to be recent transitions to multicellularity. So those green bars here display multicellular taxa. If we summarize this, it would mean that in proteobacteria and bacteroidetes, they seem to be recent transitions, whereas Cyanobacteria, Chloroflexi, and Actinobacteria seem to have a multicellular character set for much longer in Earth history. So fossils we find from the Precambrian, early Archean and early Proterozoic, they're more likely to belong to any of those groups among the living taxa. As for the gunflint shirt, where we said those large filaments, they could resemble Cyanobacteria or Proteobacteria. It seems that they're much size-wise, they compare to Cyanobacteria, and phylogenetically, it seems that Cyanobacteria have been present at that time at least. So now, well, what do we know about modern Cyanobacteria? I already told you that they show quite a variety of different morphotypes. If we now look at Cyanobacteria, multicellular Cyanobacteria today, we see, we see quite a variety of different taxa here, also a unicellular one, and we see that those multicellular features, they're quite different actually. They might not reach complexity levels as we see in plants or animals, but they still share basic multicellular features, which you will find also in other multicellular taxa, such as cell-to-cell adhesion, directional growth, an extracellular sheath, which will enable a communication of cells over longer distances, Necredian cells, which are cells that die off basically via apoptosis. The filament can divide at those spots. And some simple forms of terminal cell differentiation. So those are so-called heterocysts. Here's a filament of Anabena, and we see those are vegetative cells. Here we have photosynthesis, oxygenic photosynthesis. In those heterocysts, they are colored more. They have a more brownish color. That's because they fix nitrogen. The enzyme nitrogenase that fixes nitrogen is very sensitive to oxygen, so they undergo strong morphological changes. This is sort of the cell differentiates to do solely nitrogen fixation, no photosynthesis anymore, and this is a terminal differentiation, so it can't go back. This cell won't be able to divide anymore. It will be a heterocyst for the rest of its life. So it's sort of already a simple form of complexity, a division of labor in those simple prokaryotes. Cyanobacteria can also form acunids. Those are resting cells that will help them to sustain for very long periods if conditions are not as good. And this is just, so this is summarized, and according to those differences, they have been summarized into 5 morphological sections. So I'm going to go on to tell you about phylogenomic data and how I reconstructed evolutionary histories of cyanobacteria using phylogenomic data. So for this, please keep in mind this little cartoon here is going to reappear, and also the color code is going to reappear. So we do have unicellular cyanobacteria up here in yellow and orange. And we do have simple multicellular forms here in green, and we do have multicellular forms with cell differentiation, heterocysts, and acinids, and even branching patterns down here in purple and blue. Now, how did those morphotypes evolve? This is a phylogeny I have reconstructed based on 16S gene, 16S ribosomal RNA. Those are 1,220 cyanobacterial taxa. This is basically all we know about cyanobacteria. And what you see immediately is it's nothing that you would expect, like this traditional transition. You have a unicellular cyanobacterium, it becomes multicellular, it gains some terminal differentiation, it becomes more complex. But instead we see that multicellularity is scattered all across the phylogeny. The same for unicellularity. We do have unicellularity here is another one. The only thing that seems to cluster a bit is this cell differentiation. So they seem to form a sort of more monophyletic group. Here will be the output, by the way. Again, the problem is with such a large data set and only one gene that you have almost no support at those regions, or no support at all. So it's very hard to reconstruct ancient evolution of cyanobacteria on this phylogeny. So I did subsample from this huge phylogeny, This is a phylogenomic tree. It's based on 755 genes. It has way more information. Some of those taxa I sequenced myself. The others have recently been sequenced by the Pasteur Culture Collection, with whom I collaborate. And oh, no, I'm taking everything off. So I did, again, character state reconstruction on this huge phylogeny. Again, it's the same as before. So we have black will be a multicellular character state, yellow will be a unicellular character state. You see here the different colors denote, again, the different morphotypes. We see multicellularity is scattered across the phylogeny, unicellularity is scattered across the phylogeny. And the outgroup would have grouped somewhere here. I did a lot of outgroup testing, so this Schirrmeister seems to be closest to the outgroup. If we reconstruct the evolution of multicellularity across this phylogeny, this is what we get. So we see the origin cannot be resolved 100%, but then very early on we have this ancient multicellular lineage evolving in Cyanobacteria. And apparently, multicellularity, it gets lost several times within the cyanobacteria. So at least 5 times we see that this ancient multicellular character state has been lost again. So most of the unicellular taxa we see today actually lost a previous multicellular character state to adapt to a new environment. And within one of those unicellular clades, we even see reversals to multicellularity. So recent transitions where cyanobacteria evolved to multicellular character state again. This is in those results I just showed. They do agree with a study I conducted previously based on only one gene. Based on only one gene here on 16S ribosomal RNA, where we also gained a really ancient multicellular lineage down here and several losses later on. Now the question is, we do see a lot of multicellular taxa throughout the Proterozoic that are very likely cyanobacteria. Is there a way that we could, maybe by using divergence time estimation, using some fossils as calibration points, could we reconstruct the evolution of those of multicellularity in cyanobacteria? The question would be, we know cyanobacteria have been around at the Great Oxidation Event. We do see in the Gunflint very likely we do have multicellular taxa. We do see a lot of other cyanobacterial multicellular taxa later on in the Proterozoic. So the question is, when did multicellularity evolve? Could it be related to the Great Oxidation Event? Could multicellularity have been some advantage helping cyanobacteria to become more abundant, therefore produce more oxygen, causing this increase of oxygen that suddenly occurs at the end of the Archean? Or is it more likely that we have an increase of oxygen and multicellularity evolving as a sort of adaptation to new environments that are suddenly occurring after the Great Oxidation Event? I'm trying to test, I did try to test those 2 hypotheses. So for that, I did calibrate my phylogeny, the one I showed you before. Various calibrations. So first of all, the root. What do we know about the origin of cyanobacteria? We know they have been around at the Great Oxidation Event, probably even earlier, we know now. And we know they probably, they very likely couldn't exist before in the Hadean Eon because their temperatures would have been too high. We have those heavy bombardments. So we could assume that life didn't exist at this point. So we can restrain the root to that cyanobacteria occurred sometime in between those 2 dates. Probably more towards the Great Oxidation Event because otherwise we would observe oxygen increases already earlier. So this is exponential distribution. Actinides, I told you about the resting cell cyanobacteria form. They are formed by those multicellular cyanobacteria. Sorry, I think it stopped. Can you hear me? Is that okay? Okay, sorry, I think the microphone stopped. Okay, so so cyanobacteria, the actinides, they are formed by multicellular cyanobacteria that can do terminal cell differentiation. I showed you they seem to cluster, they seem to be a monophyletic group. So we can use this information. We do have fossil actinides around 2.1 billion years ago. And they do, so given that this group seems to be monophyletic, we can use those actinides to calibrate this origin of this group with terminal cell differentiation. So we know they must have been around at the time when we see this fossil, which is 2.1 billion years ago. Likely they occurred, they evolved sometime before. They do have the heterocysts, which protect the nitrogenase from oxygen. So it's rather likely that they evolved after the Great Oxidation Event as a response to increasing oxygen, forming those heterocysts. So if we we can restrain this date there. Now I want to test those two hypotheses. So basically, we know multicellularity occurred very, very early in the phylogeny. We can now restrain the age of this multicellular ancestor to be force it to be more likely before the Great Oxidation Event or more likely after the Great Oxidation Event. So if I use those two different hypotheses as an input to calibrate my phylogeny, this is, so here again a summary of the different calibrations, so the root, those terminally differentiated cyanobacteria and those two hypotheses, we come to the results. So again, this is the phylogeny we had. You see again the different multicellularity scattered across the phylogeny. Here, this is based on 16S rRNA, so this group is a bit less well resolved than with the phylogenomic tree, but they are still very similar. Now, if we only look at the evolution of multicellularity, so unicellular and multicellular taxa, unicellular in yellow, green, multicellular, and we remember that we have this very early transition to multicellularity in cyanobacteria. And now we look at our two hypotheses. So, hypothesis that we have multicellularity evolving before the Great Oxidation Event, maybe being causal to the Great Oxidation Event, or the second hypothesis that we have multicellularity evolving after the Great Oxidation Event, sort of as a response, as an adaptation to new habitats after this increase of oxygen. We get those results. So, hypothesis 1 in yellow, hypothesis 2 in blue. And those are median node ages, and there's quite a huge difference over millions of years, but yet they all point rather to a very early origin of cyanobacteria, somewhere in the Archean Aeon. So they do have, we can still gain some information out of those different calibration tests. If we look at the evolution of multicellularity, it seems that multicellularity, both hypotheses, even the one where we assumed that multicellularity would evolve after the Great Oxidation Event, where we basically put data in our analysis that should have forced this node to occur later on. Yet still, it seems more likely that multicellularity occurred before the Great Oxidation Event or towards this increase of oxygen. So maybe multicellularity had some advantages indeed and helped cyanobacteria to become more abundant, therefore produce more oxygen. The most recent common ancestors of all the other living cyanobacterial taxa, they occur after the Great Oxidation Event. So if we compare this now to the phylogenomic data I had, so this is the phylogenomic tree, this is the tree I just showed you. Oh, yeah. So what we see here, this is the node where we have multicellularity occurring. It seems to be even pushed further back in time in this tree, so in the phylogenomic tree, which I'm going to do the divergence time estimation again with this tree, where we have way more information, 755 genes compared to only one gene. But it could be that it will push the date even further back. I will have a look. To summarize what I just told you, so I tried to make this little cartoon. I hope it's not too bad. It's just a schematic, so and this is supposed to be a stromatolite. I'm sorry, I wanted to point it out, but I hope you can still see it somehow. So if we assume that we have, in the Archean Aeon, we have maybe some microbial mats, we have planktonic forms, and then we have a unicellular cyanobacteria occurring. The problem here being that at the same time, during the Archean, we don't have an ozone layer, so UV might have been very lethal for those bacteria. So they might have had a problem there because they need light energy to produce, to do photosynthesis, to gain energy, but at the same time they have to protect themselves or to go deeper down in sediments to hide from the UVC that is still around at that time. So if we look at multicellular cyanobacteria, when they occur, they might have the advantage, first of all, they have motility, they can move around. So they have the advantage that they can move within a bacterial mat to try to hide from lethal UV portions. Second, a second mechanism that could have been very advantageous is attachment. They have a larger surface, they can easily attach and grow new mats. So those two things might have been great advantages at this point. Once we have, this might have helped them to become more abundant, increase oxygen, we get this increase of oxygen at the Great Oxidation Event, and then later on, this will help other mat communities later on to grow in photosynthetic environments, because after the Great Oxidation Event, we have an ozone layer, we don't have those lethal UV radiation anymore. So later on, we do see, at least in the gunflint chode, we do see a huge variety of morphotypes that appear, and I'm not aware of any fossil deposits before the Great Oxidation Event that shows such a variety of different morphotypes. So this is just an idea how this could have functioned. I would like to thank a lot of collaborators and colleagues at the University of Bristol, Phil Donoghue, Muriel Booker from the Pasteur Culture Collection that helps me with the DNA, that sent me some DNA of cyanobacteria, Joachim Reitner from the University of Göttingen, and some collaborators, Alexandra Antonelli and Julian De Vos, with whom I worked in Zurich at the University of Zurich, Michael Brazier from the University of Oxford, Francis Westall, and various other people that helped me. A lot of people from Bristol, obviously, and some funding bodies that supported my work. And thank you.